The neuron consists of dendrites, nucleus, cell body or soma, axon, axon hillock, synapse, and synaptic terminals. The first neuron is called the presynaptic neuron, and the second neuron is the postsynaptic one. Neurons carry action potential, the change in electrical potential, associated with the passage of an impulse along the membrane of a nerve cell from the dendrites through the axon and to the synaptic terminal. Once there, the impulse is transferred across the synapse and to the dendrites of the next neuron. Up close, the phospholipid membrane of a neuron has embedded channel proteins. These proteins allow for the transfer of sodium and potassium ions. If the channels are open, the ions will move with their concentration gradients. Sodium moves into the cell and potassium moves out. Ions can move against their concentration gradient with the use of ATP and a sodium-potassium pump. The sodium-potassium pump moves 3 sodium ions out for every 2 potassium ions it pumps into the cell. The sodium-potassium pump is useful for resetting the resting state of the neuron after the action potential is passed. During resting state, the inside of the cell is negative and the outside of the cell is positive.
The generation of action potential occurs in several stages: resting potential, depolarization, rising, falling, undershoot, and then returning to resting potential. At a neuron's resting state, the inside of the cell is negatively charged while the outside of the cell has a positive charge. This is due to the distribution of ions across the membrane. The membrane potential of a resting neuron is 70 millivolts. A stimulus causes some of the sodium channels to open, allowing for the movement of sodium ions into the cell. This is called depolarization.
Once the membrane potential has reached the threshold of negative 55 millivolts, the voltage-gated sodium channels in the area open, allowing more sodium to flow into the cell. There's a chain reaction caused by the flow of sodium ions where the open channels make the channels near them open in response to the change in voltage. This continues to increase the membrane potential. When the membrane potential reaches positive 35 millivolts, the voltage-gated sodium channels close and the voltage-gated potassium channels open, allowing potassium ions to flow down their concentration gradient. The flow of potassium out of the cell lowers the membrane potential in a process called repolarization.
The membrane potential reaches negative 80 millivolts at the end of the falling phase. At this point, some of the voltage-gated potassium channels close, although not all. The sodium-potassium pump works to rebalance the ion concentrations, pumping sodium out of the cell and potassium into it. Once the sodium-potassium ion pump has rebalanced the ion concentrations, the membrane potential returns to negative 70 millivolts. The neuron is ready to receive another signal.
Tetrodotoxin is a sodium channel blocker which binds to the channel, preventing any passage of sodium ions. This stops the membrane potential from ever reaching the threshold, making it so that no signal can be sent through the neuron. Symptoms of poisoning in the early stages are mouth and facial numbness. The muscles become paralyzed, including respiratory muscles, which leads to respiratory paralysis and ultimately death.